My name is Hiroyoshi Aoki, TOYO Corporation. Thank you for attending my webinar today. Earlier, TOYO launched agarose gel microcapsules for the single-cell genome analysis of microorganisms. The AGMs can be easily prepared. They provide an enabling environment for picoliter scale gene amplification to obtain high-precision single-cell genome analysis data. Our AGM Reagent Kit, which was based on the technology, was approved for marketing and has been commercially available. It is now used by many users. In this webinar, I will discuss the technological details of the AGM Reagent Kit. First, I will briefly present my career history. When I was a student, I had already cloned enzyme genes capable of decomposing polysaccharides from molds and constructed mass expression systems. This image shows a three-dimensional view of the enzyme. I discovered that the common motif was the beta sheet, on which clefts are formed to decompose polysaccharides. After graduating from the doctoral program, I was involved in rice genome analysis at a research institute. Rice genome sizes are as large as 390 mbp. When I worked on the rice genome, next-generation sequencers had not yet been available, therefore, genomic analysis was extremely difficult. The international projects involving Japan and many other countries, however, worked on the difficult task and succeeded in decoding the rice genome. Later I joined with a company and was involved in the development of microfluidic chips and reaction detection devices, which are technologies capable of simultaneously determining multiple infections from minute amounts of mouse blood. More recently, I worked in Reagent and then I joined TOYO Corporation this year. In this webinar, I will discuss agarose gel microcapsule, or AGM technology, as well as the background, characteristics, and overview of AGM development. I will also report demonstration experiments in E. coli and human gut microbiota, mock sample, and actual samples of the termite gut microbiota. Next, I will introduce the AGM Reagent Kit. We will present a brief description of the kit components, required devices, working hours required, and where to purchase the kit. First, I will explain AGM technology. Countless microorganisms inhabit our environment, and they are deeply connected to human health, the environment, and industry. However, culturable microorganisms account for only a small fraction of all the microorganisms, and the majority of them are known as uncultivated microorganisms. The uncultivated microorganisms are also known as microbial dark matter because their functions are unknown. Recently, however, genomic DNA analysis of uncultivated microorganisms has advanced, and their functions have been inferred from their genes. They have been gradually characterized. Conventionally, genomic analysis of uncultivated microorganisms mainly relied on the metagenomic approach. In metagenomic analysis, genomic DNA is extracted from environmental microorganisms and analyzed with a DNA sequencer to obtain sequence reads or short genomic fragments. The sequence reads are assembled on the basis of mutual homology to create large contigs. Then, genes are identified from the genome sequences of the contigs, and their functions are inferred. The workflow is known as annotation. However, since metagenomics is the genomic analysis of a population of microorganisms, it is difficult to sequence whole genomes. It may wrongly bring together genomic DNAs from different microorganisms, and it is difficult to localize intracellular plasmid DNAs to specific genomic DNA, because they exist separately from genomes. Single-cell genomics technology, which directly isolates and analyzes microorganisms, address these issues. In single-cell genome analysis, a small amount of isolated microbial genomic DNA is amplified by multiple displacement amplification reactions using Phi29 DNA polymerase, and then genome analysis is performed in the same way. The genome sequences delivered by single-cell genome analysis are all derived from a single microbial cell, therefore, the technology usefully serves for analysis of microorganisms localized in specific environment and as reference sequences in metagenomic analysis. However, single-cell genomics had been challenged by local amplification bias in MDA, which would complicate the ability to obtain information about the whole genome. To date, attempts have been made to reduce the reaction mixture in volume to reduce the amplification bias. A method of passing water in oil droplets in microchannels using MEMS technology and MDA in agarose gel beads has been reported as attempts to reduce the amount of the reaction mixture. This is the result of adding a single E. coli cell to the microchannel, performing MDA within a PL scale droplet, and then performing DNA sequencing. In the graphs on the right, the horizontal axes indicate genomic positions, and the vertical axes indicate the logarithm of the relative number of sequences at different genome sites. Conventional 30 microliters bulk MDA cover different numbers of sequences depending on the genomic positions, which means that sequences varied widely. On the other hand, the MDA reaction in as small as a 95 pl droplet delivered uniform amplification throughout the genome. Conventionally, MDA reacts in a liquid with several tens of microliter, but in this test, amplification bias was inhibited by allowing the reaction in a picoliter order. Various microorganisms exist in the experimental environment. Therefore, the experimental equipment and containers are sterilized and not reused to prevent the microorganisms from complicating single-cell microbial genome analysis. However, sterilization of the MEMS devices is difficult. It is also a time-consuming task for ordinary microbial researchers to assemble the devices. In addition, it is difficult to dispose of the devices. To address the problems, we developed a more practical and simple micro-reaction vessels, agarose gel microcapsules, which contain liquid within the agarose gel shell. An AGM consists of a core made of liquid alginate sol and an agarose shell gel containing the core. Alginic acid and agarose are both natural polymers obtained from seaweed and can be easily gelled and liquefied. This slide shows the agarose shell surface observed using a surface scanning electron microscope. The shell has a pore diameter of several tens of nanometers, and proteins of several nanometers permeate it. On the other hand, as for linear DNAs, calculations show that oligo-DNA passes through the shell, while longer or several kilobase long DNA sequences cannot penetrate the shell. It means that when MDA is performed inside an AGM, reaction reagents, such as primer DNA and enzymes, permeate the AGM, while DNA sequences amplified by MDA, dendritic polymers, are prevented from leaking through the AGM and outside microorganisms, and the DNA sequences are expected to be blocked. The AGMs can be easily prepared. First, disperse a core gelling agent made of microorganisms and alginic acid and a suspension of gelling agent A in oil. Then, add gelling agent B to the mixture and gel the alginic acid with liberated calcium to prepare an alginic acid core. Next, wash the alginate core and mix the core with the shell gelling agent made of agarose. Then, suspend and gel the mixture in oil. After washing the gel, liquefy the core using a core sol agent. The procedure results in AGM capable of embedding microorganisms. What was most difficult in developing AGM was to keep the gelled agarose shell stable. A simple mixture of agarose and oil will immediately separate into upper and lower layers because their specific gravities differ and will never form stable shell. Therefore, we attempted to gel the agarose shell using oil that has approximately the same specific gravity as water. The photos in the upper half show the mixtures of water and the oil. The oil allows emulsion to be maintained for a long time. When the oil was used to gel agarose gel beads, the oil prevented gel sedimentation and aggregation and achieved high yields. To demonstrate our AGM product, we performed single-cell genome analysis using AGM with Escherichia coli, a mock sample of a mixture containing 15 types of human gut bacteria and, as an actual environmental sample, termite gut microbiota. In particular, termite gut microbes are uncultivated microorganisms. Are mainly microorganisms with high cellulolytic activity. And they have well-developed symbiotic relationships with eukaryotic endosymbiotic bacteria and are microbiologically interesting. First, we serially diluted the bacterial cells and optimized the bacterial cell concentration so that there was less than one bacterial cell per core. This resulted from adding serially diluted E. coli to alginic acid to create a core. E. coli cells were stained with cyber green eye. The phase contrast image is shown in red, and the fluorescence image is shown in green. When 3 by 10 to the power of 6 E. coli were added to 1 milliliter of alginate mixture, many alginate cores containing a single bacterial cell were obtained. In other words, when you prepare AGMs by yourself, you should ensure that the amount of the bacteria to be added is no higher than the level in order to produce AGMs containing a single cell. Next, we created an AGM library. Although there was some variation because it was prepared by stirring by hand, the resulting AGM library contained microcapsules with a core diameter of approximately several tens of micrometers and a core volume of approximately 10 to 20 pl. Approximately 500,000 AGMs containing bacterial cells were obtained in one preparation round, of which the AGMs containing a single cell accounted for more than 90%. Once alkaline denaturated, genomic DNA sequences were amplified by multiple displacement amplification method. After the reaction, the amplified DNA sequences were stained with cyber green eye. Because amplified DNA sequences were diffused, the cores were more filled with amplified DNA sequences than MDA and agarose gel beads, which were prepared for comparison. DNA amplification was detected in approximately 90% of AGMs containing E. coli. A micromanipulator is shown is used to isolate DNA sequences amplified by the MDA reaction in AGM. MDA and AGM amplified DNA sequences stained with cyber green were aspirated through a fine glass tube under an inverted fluorescence phase contrast microscope. They were dispensed into PCR caps, and the caps were transferred to tubes. Although it requires some skill and the throughput is low, it can reliably isolate approximately 5 samples per hour. Once MDA was finished, AGM was isolated using a micromanipulator, and the capsules were heated for dissolution. The amplified DNA sequences were subjected to 16S RNA PCR and DNA sequencing to check quality. The amplified DNA sequences then were fragmented using QI-ASIC and sequenced using me-seq. The large amount of sequence reads, which were obtained by a DNA sequencer, were assembled using these various tools to obtain single-cell amplified genomes, which are genomic data derived from a single cell. The obtained sequence reads were trimmed with adapters, de novo assembled using SP-AIDS. Contigs of 1 kilobit or more were extracted from assembled sequence reads, and taxonomically classified using GTDB-TK. These SAGs were evaluated for quality using Checkman-Kvost. Also, in order to evaluate amplification bias, we determined that the genome completeness obtained by assembling sequence reads while gradually increasing reads in number and reads per 50 kilobits obtained after mapping reads to the reference genome. We evaluated the uniformity of their distribution using the Lorentz curve and Gini coefficient. The graphs show the comparison in genome completeness and number of contigs between MDA and AGM and FAX-MDA when a different number of reads were assembled. MDA reaction in AGMs, or MDA in AGM, was compared with conventional FAX isolation and microliter scale MDA reaction, or FAX-MDA process, in the coverage rate and the number of contigs using reads whose total sequence length is equivalent to 2 to 60 times the genome length. The assembly results showed that completeness increased slowly as the number of reads increased in the FAX-MDA reactions, whereas completeness increased rapidly and reached saturation at 40 to 60x coverage with the MDA and AGM reaction. As for the number of contigs, when genomes were amplified with the conventional FAX-MDA method, the number of contigs did not almost change even when the coverage increased. When genomes were amplified with the MDA and AGM method, contigs noticeably decreased in number as coverage increased. For several SAGs, we compared heat maps for the number of reads between AGM and the conventional method. In conventional FAX-MDA, regions with a sequencing depth that was low at the beginning remained low even when coverage increased. When genomes were amplified by MDA and AGM, as coverage increased, reads were gradually dispersed, and uniform distribution was achieved. Also, visual inspection found no characteristics typical to areas where coverage increased easily or areas where it did not increase easily. Next, the amplified genomes were evaluated for amplification bias using the Lorentz curve and Gini coefficient. The Lorentz curve and Gini coefficient are commonly used in sociology studies to assess wealth homogeneity, but they are also used to assess amplification bias in single-cell genomes. First, the sequence depth, which is the average number of reads per base, was determined for every 50 kilobits, and then the sequence depths were plotted on the horizontal axis, and the total sequence depth at that point was plotted on the vertical axis. The plot generally draws a curve that bows down toward right like this. This is known as the Lorentz curve. If the distribution is uniform, a straight line is drawn and inclined at 45 degrees like this. The line is known as an even distribution line. If the area between the equal distribution line and the Lorentz curve is A and the area under the Lorentz curve is B, the Gini coefficient is defined as A/A plus B. In other words, when the read distribution is uniform, and the amplification bias is low, the curvature of the Lorentz curve is small and the Gini coefficient is also small. On the other hand, when the amplification bias is high, the curvature of the Lorentz curve and the Gini coefficient become large. Among the samples, Escherichia coli, Bacteroides, and Parabacteroids, which have a large amount of data and whose genome sequences are known, were selected to compare their Lorentz curves. In each of the populations, the SAG with a Gini coefficient close to the median value is plotted. For all the bacteria, uniformity and read distribution was remarkably higher in MDA and AGM than in the conventional method. Next, amplification biases were compared for SAGs using Gini coefficients between the two methods in the microbial populations. The numbers in parentheses indicate the numbers of data. The Gini coefficient was significantly lower in MDA and AGM method than in FAX-MDA method. More than one strain achieved more uniformly amplified DNAs in MDA and AGM method. We performed single-cell genome analysis of mock bacterial community using the MDA and AGM method. For comparison, we also conducted a similar experiment using the FAX-MDA method. Mock communities differed in cell concentrations because they were prepared simply by mixing different culture solutions, making it difficult to detect and analyze all the bacteria. However, the MDA and AGM method again allowed bacteria to be isolated from the bacterial mixture using AGMs and to be analyzed. Next, we also performed single-cell genome analysis of termite gut microorganisms using the MDA and AGM method and obtained SAGs with high completeness for these microorganisms. Among these, Endomicrobia is a cellulolytic eukaryotic endosymbiotic bacterium that lives in the termite gut. It is bacteriologically interesting, for example in the aspect of symbiotic relationship. Also, its genome sequence has been established. Therefore, we performed additional single-cell genome analysis using MDA and AGM as a benchmark and obtained SAGs with high completeness. SAGs obtained by MDA and AGM and conventional FACS-MDA methods from E. coli, mock samples, and termite gut bacteria in the demonstration experiment were compared for completeness. The comparison showed that remarkably higher completeness was achieved with the MDA and AGM method than with the conventional method in all samples. Because of its simplicity, AGM has a potential for use in extensive applications. This slide shows hybridization of MDA and AGM amplified DNA genomes with a fluorescently labeled DNA probe. The fluorescent probe was a Cy3 labeled oligonucleotide DNA, which was designed based on the E. coli 16S RNA sequence. As a result, we obtained this specific reaction. Therefore, it is possible to isolate SAGs obtained by MDA and AGM containing the desired DNA sequence from a large number of MDA and AGM libraries. The slides show bacteria cultured inside AGM. When E. coli is cultured in AGM, the bacterial cells are fully dispersed within the core and grow in the form of a ball like this. The bacteria cultured in AGMs have advantages. Being easily retained by gel cores, within which the bacteria diffuse, while bacterial cells leak out of gel beads at the portion where they are embedded. Being easily collected. The MDA and AGM method may be applied to analyze interactions between co-cultured microorganisms. Next, we will talk about the AGM Reagent Kit. This is the AGM Reagent Kit. Our company, TOYO Corporation, manufactures and sells the AGM Reagent Kit under the exclusive patent license granted from RIKEN. The kit includes the reagents and equipment needed to create an AGM library 3 times, and you may create AGMs that embeds approximately 500,000 cells in one round. There are 2 types of reagents. One refers to reagents storable at room temperature and the other refers to reagents that have to be refrigerated. For purchasing AGM reagent kits, please contact us via our website, email, or by telephone. You can also purchase from your familiar retailers. Please contact us for information. The slide provides a list of the consumables and experimental equipment needed to prepare AGMs. Basically, common sterilized plastic instruments are acceptable. Custom-made experimental equipment is not required to prepare AGMs, but commercially available equipment is sufficient. To prepare AGMs, you form an emulsion in a 50 ml tube, gel the immersion, and collect AGMs by centrifugation. We recommend a swing rotor centrifuge because it allows AGMs to be easily collected, although an angled rotor is also acceptable. In addition, make a heat block for 1.5 and 50 ml tubes available. In addition, after the AGM is prepared, bacterial cells and amplified DNA are stained with cybergreen eye for observation. Therefore, you need to use a fluorescence phase contrast microscope with blue, B, excitation, which can detect cybergreen eye. Use of a micromanipulator is recommended to isolate AGMs after amplification. After isolation, dispense them into a qPCR flat cap. You will easily observe AGMs under a microscope. Although this will partially overlap with what I discussed earlier, I would like to provide an overview of single-cell genome analysis of environmental samples using the AGM Reagent Kit. First, disperse the environmental sample in PBS. After centrifugation, collect the supernatant. To degrade contaminating DNA, treat the supernatant with DNase-I and then store it frozen. Measure the bacterial cell density and prepare an AGM library by embedding 1 microorganism cell in each gel microcapsule using the AGM Reagent Kit. AGM libraries can be created in approximately 1 to 2 days. After examining the microorganisms in the prepared AGMs using a fluorescence microscope, store it at 4C or 80C under 40% ETO for long-term storage. Our studies showed that AGMs can be stored without degradation in that condition for 1.5 years. Then, perform MDA using Repli G Ultra Fast Mini Kit and create an MDA and AGM library. Next, isolate AGMs containing amplified DNA from the MDA and AGM library using a micromanipulator. Depending on the number of analyses, it takes approximately 1 or 2 hours to isolate 10 samples. Heat the isolated AGMs to dissolve their shells and heating and determine their sequences by 16S RNA PCR and Sanger DNA sequencing. Then, prepare a sequencing library using the QI-ASIC-FX DNA library kit and analyze the library using a DNA sequencer. Roughly, the following sequence amount is required for the MDA and AGM method. Our results on E. coli amplified with the MDA and AGM method showed that a depth of 40x to 60x was the point at which the coverage rate reached saturation compared with genomes. Sequencing was performed with sequencer ME-seq in one run with read lengths up to 300 BP paired end. The genome size of E. coli was 4.6 megabits. In other words, when largely 50 coverages are targeted, 7.7 by 10.5 reads are required. In terms of data size, 50x coverages of genomes, or 230 megabits will be required. Conclusion. We launched an agarose gel microcapsule consisting of an alginate sol core and an agarose gel shell for single-cell genome analysis. AGM suppressed amplification bias by 1 round of the PL scale MDA. The AGM Reagent Kit allows many researchers to perform simple and high-quality single-cell genomic amplification. The kit will surely accelerate genomic analysis of uncultured microorganisms. That's all. Thank you for listening.